Hi, welcome to my revision video on the sliding filament model. So we're going to start by having a look at the structure of muscles. We're going to look at a skeletal muscle here, or striated muscle. We'll talk about the striations in a bit, and we're going to look at the structure of this muscle. There are many muscle fibers. These are the actual cells, the muscle cells. And we can see here one that's been pulled out, and there are many within this muscle here. So the muscle fiber, we can often see many nucleuses in the muscle fiber because they are cells that have joined during development. So embryonic development, those cells join to create long muscle fibers, and they are considered one cell, although they will have many nucleuses in there. What else can we see here? We can see an axon. So we've got an axon joining to the muscle fiber. And we've got a motor end plate here. So this is obviously bringing in a nervous impulse, and we'll talk about that in a bit as well. Obviously loads of blood capillaries here, which are bringing muscle oxygen to the muscles. And within the muscle fiber, we have many of these, which are myofibrils. And within the myofibrils, we've got myofilaments. There's a lot of myos here. And the myofilaments contain the proteins myosin and actin. And we're going to look a lot more detail about these myofilaments in a bit. The muscle fiber or muscle cell will have a plasma membrane around the outside. We call this a sarcolemma. So the plasma membrane is the same structure as any plasma membrane you've already discussed in cell biology, but this is called a sarcolemma. We've also got endoplasmic reticulum in the muscle cell, which we call sarcoplasmic reticulum. So it's a special adapted type of endoplasmic reticulum. And this one's called sarcoplasmic reticulum. And again, we'll pick up on this in a bit. The cytoplasm of the muscle fiber is called the sarcoplasm. That's a little easier to remember. So the cytoplasm is the sarcoplasm. So there's a lot of sarcos here. You need to know the definitions. And as we go on, you will pick that up. I've zoomed in here on the myofilaments. So I said it's a collective term for the myosin and the actin. The myosin filament is obvious because of these globular heads that it has. Okay, they're on a little stalk here that's hinged. So this is the myosin and the myosin heads. The myosin heads have a binding site for the actin, which we'll speak about in a second, but they also have a binding site for ATP, which again we'll pick up in a bit. We've got the actin, which is like the pearl necklace running along here. It's two polypeptide chains wrapped around each other, and that is the actin along the middle here. We've got tropomyosin, which is another protein around the outside of the actin. So that's the tropomyosin, which looks like a bit of spaghetti there. And we've also got the troponin, which is another protein found within the actin and the tropomyosin. Functions of those we will talk about in a bit. I've zoomed in here and we can see that on the actin we have myosin binding sites. So the myosin head here, the globular head here, should be able to bind under normal circumstances with the actin. So the tropomyosin, this spaghetti-like protein, blocks the actin-myosin binding site. And the tropomyosin is held in place with the troponin. So this protein here, the green protein coloured here, green protein, troponin holds the tropomyosin in place. So in a resting state, the tropomyosin blocks the actin-myosin binding site. In the presence of calcium ions, the troponin will change shape and the protein will turn. And I use the term turn specifically because tropo, as in troponin, tropo comes from the Greek tropos, which means turn, to turn. So if you remember phototropism, the plant is turning towards the sun. Okay, so troponin means turn. In the presence of calcium ions, the troponin will move and turn, and what that does is it slides the tropomyosin out of the way, and it will expose the actin, and it will expose the actin-myosin binding site. How the calcium get there? Well, we need to talk a little bit more about the structure of the muscle fiber, and we'll have a look at that now. So if you remember, this is the muscle cell, or the muscle fiber, and around the outside we have the membrane, the plasma membrane, which we call the sarcolemma. The sarcolemma acts like most membranes in most cells, and if you think about neurons, and action potential neurons, okay, like most membranes, it maintains a membrane potential. So there is a resting potential across that plasma membrane, okay? If we have an action potential, then it will move across that plasma membrane just as it does along the nerve cell membranes. At points along the sarcolemma, we've got these invaginations of the plasma membrane, and they're called T tubules. So they look like T's, okay? And the plasma membrane moves or is invaginated into the actual cell. So here we can see one of the T tubules. So here's the sarcolemma, and we've got the invagination into the actual cell. And we can see if an action potential moves along the plasma membrane or the sarcolemma, it will move down into the T tubule. So what this does is it takes the action potential into the cell. This causes something. So this causes the sarcoplasmic reticulum. If you remember, I referred to it at the beginning. It's a modified endoplasmic reticulum. And this sarcoplasmic reticulum, under these conditions, will release calcium ions. Okay, so we've got an action potential stimulating the release of calcium ions from the sarcoplasmic reticulum, and those calcium ions will diffuse across through the sarcoplasm towards the myofilament. So in the presence of calcium ions, we have the troponin moving or turning and exposing the binding site. So myosin can now bind to the actin. So the myosin head now bound to the actin will drive the actin filaments forwards. Okay, it's like this power stroke. It drives the actin forwards. At the end of this myosin head power stroke, something happens. The ATP will then bind to the head of the myosin. Okay, that has two functions. One, it sets up the next round, okay, in terms of energy, and we'll deal with that in a second. But the other thing is, as it's bound now, the myosin head is now bound to ATP, it's no longer bound to the actin filament, the actin binding site. So this causes the myosin head to release from the actin. At this point, the ATP is hydrolyzed to ADP. So here we can see the ADP still attached to the myosin head. The hydrolysis of ATP into ADP causes the myosin head to move. It gives the energy required to move or change the structure of this protein so that it is now moving backwards. I can't think of many other terms here, but it's almost like cocking a gun. So the hydrolysis of ATP causes the myosin to move into this cocked position, and it's poised, ready to go. So it's almost like cocking of a gun. So the myosin head will now bind to the actin further back along the actin filament. So the energy in this cocked myosin head, this sprung myosin head, is then obviously released into this power stroke we saw before, that drives the actin molecule, the actin filament forwards. We can also see here that the ADP is now released. Now the cycle continues. So I will move back to the idea of ATP rebinding. Okay, the myosin head releasing from the actin again, and the myosin head moving back into the cocked position. So this is muscle contraction, and the muscle contraction is causing the actin filament to move forward, and the myosin head, this, the energy from the ATP within the cot position of the myosin head does that power stroke forward. So we're now going to look at the striations in this muscle fibre up here and how these come about. To do this, we need to again delve a little deeper into the structure of the actin and the myosin. So we've got the muscle fibre up here, the muscle cell. We've got the myofibril here, and I've pulled a myofibril out there, and within that myofibril we can see a graphical representation of the myosin. Here it looks like a bubbly cucumber, and we've got the actin, which is the two polypeptide chains here in red, and we've also got this blue structure here, which is cross-linked actin. So this is what's holding the whole thing together, really. If we look at the dark striation here, so the dark stripe up here, if we look at what's happening and what causes that, well, this is the dark band here. So in the graphical representation here, we've got the dark band. This would be the dark stripe here. And why does it look dark? Well, it looks dark because you have a lot of protein going on here. So if I was to shine a light through here, it's got to get through actin filaments, it's got to get through myosin filaments, and it doesn't. So in which case it's going to look dark, okay? The observant among you might notice the bit in the middle here where there is a lack of actin, obviously, and you might consider that to be a little lighter, and that is true. So within the dark band, we have a lighter region. We call this the H zone, okay? So that's the H zone, so it sits within the middle of this dark band. Okay, so now we're going to look at the light bands here, the light regions of this striated muscle. So what's going on here? So these are the light, we call them, these are light bands, okay? Now, clearly all of this would be a light band. It's all going to look light, but you'll see why I'm just labeling it from this point to here in a bit, okay? So we've got a light band here. Why is it light? Well, there's no myosin, okay? So it's going to look lighter. Okay, one last thing is the Z line. So the Z line is this very distinct line, right? So it's a small line that runs through. It will look darker because you've got the cross-linking across the actin filaments. Up here you can see it looks a bit like a Z, so that's how I remember it. So the Z line passes down here. Now, because it's so distinct and because it's so clear, it's a really nice point to take a measurement from. Okay, so we can measure it from Z line to Z line, okay? And that gives us a value for the length of a particular region, okay? And that looks like this. And we call this the sarcomere. So it's the distance between two Z lines, okay? And as I said, it's a really handy way of measuring the relative size of the myofibril, the relative size of the myofilament, and also the relative size of the muscle, okay? So when it's contracted, we can measure the size of a muscle by looking at the length of this sarcomere, so between Z lines. So in this slide, we can see a relaxed muscle, and we can also see a contracted muscle. If you remember, obviously the myosin filaments here and the actin filaments here in the relaxed state, and then we've got the contracted state. So that's obviously the bit between the sliding filament model where the myosin drags the actin along, okay? So if we look at the H zone in a relaxed muscle and compare it to a contracted muscle, we see the H zone decreasing in length. Okay, if you remember, the H zone is a lighter region within the dark band. Over here we can see the H zone here, okay, a lighter region within the dark band, and in a contracted muscle, that H zone gets shorter. Now the dark band doesn't actually change length at all, okay? The myosin filaments will remain the same length, okay? And in a contracted muscle, they're the same length, okay? It's the actin that's moving, so the myosin stays the same. So our overall dark band doesn't actually change in length. Okay, the H band within it changes length. However, however, the dark band does not change in length. Okay. Now, if you remember, we were talking about the light band earlier, and it was odd that I only labelled this side of the whole light area, the light region, as being the light band. The reason is this. If you remember, we measure from the Z line to the Z line, okay, which is the sarcomere. So only this side of that Z line belongs to this sarcomere, okay? That side belongs to the other one next to it, and this side belongs to the other one next to it, okay? So we've got to take a reading from the Z line. So it's only from the Z line to obviously the end of the myosin filament, which we call the light band, okay? As we move from the relaxed state to the contracted state, we see a reduction in the length of the light band, and we can see that from the actual image here of the muscle, decreases in length. Now the total length, the sarcomere in the relaxed state decreases as it moves to the contracted state. Okay, so the sarcomere reduces in length. So the distance between Z line and Z line reduces in length. Okay, that's the lot for today. Thank you for watching. I hope it's been useful. If not, give a shout and I can go through it in class.